Yeah, thank you very much for this kind introduction. Also for the invitation, originally invitation to come to India, which I would have very much liked to do. Now there is an online meeting, which I think is also very nice because that is at least a way how we can stay connected and have some scientific exchange despite all the restrictions that we face right now. So I'm very happy to be here. And I'm also very happy to see that there are so many people in the audience. So I will start right away and tell you a little bit about our work on magnetotaxis. You see already in the background of this title slide that there are bacteria here that swim, and they all swim in one direction, namely here to the right. And that's because there is a magnetic field that directs them to swim to the right. Before I really go into this more, I want to say that everything I'm telling you is the results from a collaboration, long-standing collaboration for about 10 years now between my group, which does the theoretical part, and the group of Damien Fèvre, which does the experimental part of the project. We started that while we were both at the Max Planck Institute of Colloids and Interfaces in Potsdam, but over the last years we have both moved. So I'm now at the University of Göttingen, and Damien is at the CEA Cadarache in the south of France, but the collaboration is still ongoing. Okay, magnetotaxis. Magnetotaxis in the end is an example for the motion of an organism. And if you look at nature, then you see that organisms move on all scales. You can go to whole animals or humans moving. You can go down to the cellular scale. You see bacteria swimming in that case, or cells crawling. And you can go to motion inside cells where you have molecular motors walking along cytoskeletal filaments. And in all these cases, there is motion, and very often this motion is directed in some way. So what I want to focus on today is this middle scale here, the cellular scale. They have stopped moving. Let's start them again. And specifically what you see here, namely the swimming of bacteria. And that bacteria move was discovered already very long ago, basically at the same time when bacteria were first observed by van Leeuwenhoek, who built a microscope, and under his microscope found that there are many single-celled organisms which he drew. He discovered that they come in very different shapes. There are many different types, these elongated ones, the spiral ones, the more spherical ones. And in one case, he also reported that they moved, and in one case, he even reported a trajectory. So this is probably the first bacterial trajectory that was ever recorded. I'm not sure how realistic this real path here is with these loops, but anyway, that's what he drew more than 300 years ago. Also, studying the motion of microorganisms theoretically has a long tradition. About 100 years ago, Reinhold Fürth argued that the motion of microorganisms can be described as a random walk. It's basically something like Brownian motion. What he said is, what he wrote in his paper, the title is Brownian motion under consideration of a persistence of the direction of motion. And that was his main point, that the Brownian motion of microorganisms is not a simple random walk where you just get an average no displacement and a mean square displacement that increases linearly with time, but rather it is a persistent random walk, which means something like this. They go straight for some time, but on long times, they do change their direction very often, so that on long times their motion is randomized. So you do have, again, on long times, no average displacement, but you do have a mean square displacement that on short times behaves like the time squared, because you have a velocity on short times and a fixed direction. But on long times, this gets randomized, and you get, again, a diffusive behavior where the mean square displacement is linear in time, with an effective diffusion coefficient that scales like the square of the velocity times the time over which this bacterium or other microbe moves straight. So this is called a persistent random walk. You go straight for some time, but then over longer times, the motion is randomized. That was Fürth's argument 100 years ago, and that's still the way how we describe motions of bacteria today. In fact, the minimal model that we use today is a so-called active Brownian particle. So we assume that our bacterium is a particle that has a position and it has an orientation. And the position and orientation of this bacterium follows a Langevin equation, where position is given by changes according to a velocity. This is the self-propulsion, so it moves in the direction, in its current direction. It may be subject to external forces, but it's also subject to noise. So this alone would just be Brownian motion in the position. And then the orientation, which enters in the self-propulsion here, that is also subject to potentially external torques. We'll see an example later when we have magnetic fields, but also noise. So on long times, this is randomized by noise. And you can think of this, if you forget about torques and forces, you can think of this not so much as a random walk, than as a random drive, where you drive your car and someone sits next to you and jiggles your steering wheel randomly. Don't try this, this is very dangerous, but that's the way how you can imagine how a bacterium moves. Okay, if we do this in 2 dimensions, the things get a bit simpler, because the orientation, we don't really need a vector for the orientation, we only need an angle. And then we can describe this by 3 differential equations for the position in 2D and the orientation with 1 angle. And so that's very often done because that's simpler than the full 3-dimensional description. Okay, so this is general way of describing motion of bacteria or other microorganisms as a random walk. Now we're specifically interested in the magnetotactic bacteria. Sorry, I forgot this. Before we get to magnetotactic bacteria, there's one slide here that shows you what I've shown you before, namely that these persistent random walks, they are directed on short time scales with a scaling of the mean square displacement with the square of time. And then on longer times, this gets randomized and you get this effective diffusive behavior. And what you see here for this full model that I just showed you, there's 2 more regimes. So on very short times, the translational Brownian motion will be visible. Very often this is not really relevant, but this will happen on very short times here, where basically the self-propulsion is not visible yet compared to the Brownian motion of the bacterium. And then that's the case we're in the end interested in on very long times, this random walk that you get from going into random directions. This will be biased again, for example, by a magnetic field, but it could also be biased by other things. Okay, there is a question, so let's feel free to ask questions during the talk.Maybe we can take that question now.
Okay, can you hear me?
Yes.
Okay, so in this plot, is it to scale, like, for example, where you are showing the taxis and the very long time limit, I would expect that the steep would be, the slope of that regime would be smaller, no? Compared to a typical run speed, it seems steeper. This one, yeah, the slope here seems steeper than the other regime. So that's why I would expect this to be smaller.
Okay, so 2 things here. This is a double logarithmic plot. Okay, so the slope here is...
Okay, sorry. I understand.
The slope is the exponent. And what you mean is the velocity should be smaller. And that's exactly why this curve here is shifted downwards. Okay? So in principle, you see exactly what you mean.
Okay, okay, thanks.
Okay, but maybe now we come to magnetotaxis. I've shown you this before. I show you another movie of this type. So you now see the bacteria moving. They have all moved to the right here and accumulated here at the boundary of a droplet. And now we move the magnet downwards. Let me go back. The magnet should have moved. Now the magnet moves. Okay, now we direct the bacteria to move downwards and we turn it again so they move to the left, and you see that we can really steer these bacteria. And the nice thing is that this steering mechanism works remotely. So this is interesting if you're thinking about making micro robots application or something like this. Then of course it's very important that you don't have to manually access a microscopic particle to direct it, but rather you can do it remotely, like with this magnetic field. Okay, but how do these bacteria do this? Maybe one more comment here. If you want to see much more fancy types of steering, some Dutch colleagues have made a nice video. Just Google dancing magnetotactic bacteria. You will find some way more fancy types of steering the bacteria. Do this after my presentation and not now, okay? But the question now is, how do these bacteria do this? And they do this because they have special organelles that are magnetic. And this is what you see here. So this is one of these cells. It's a species called Magnetospirillum griffiths-waldense. And you see these black particles here. The black particles are magnetic nanoparticles. And if one zooms in, which has been done with cryo-EM, then one sees that these are there are these particles. They are wrapped inside a vesicle, so there is a lipid membrane around it, and they are attached to a cytoskeletal structure, some filament here, so that you get this nice one-dimensional structure that can act like a compass needle for the cell. So what we have here is basically a permanently magnetic structure that can act like a compass needle, and if you put that into a magnetic field, that can passively orient the cell. And there's many reasons why people are interested in these bacteria. So these are bacteria that naturally make these magnetic nanoparticles. That's already one of the reasons why people are interested, because you can learn quite a bit from these bacteria on how to make magnetic nanoparticles. But of course, there is also a biology side here. People are interested in their role in the environment, in the diversity of these bacteria, in their evolution. And they're, of course, also a model for how structures form in bacteria. These are organelles in bacteria, something that maybe when I was a student was still not believed to even exist. And now that's seen that it's fairly common. And this would be one system where you could study this as a model system. And of course, there's the physics side, which is what I'm mostly interested in. There's the question, what role do the forces, these physical forces of magnetism, have in a cell? How do they act together with the biological processes? And there are also some applications in biotechnology. For example, if you have magnetic bacteria and you can take the bacteria, make the bacteria take something up, then you can extract the bacteria from the environment or from water or so with a magnet as a way of extracting whatever they have taken up, for example, toxic compounds or so. And of course, you could use the particles themselves, and you could use the bacteria to build some kind of biohybrid micro robots. Okay, but from the physics point of view, these bacteria, as I just said, they align passively with a magnetic field. They don't move because of the magnetic field, but they move because they have flagella. So they have a flagellum or two or several that they rotate, and this propels them forward. So you have a non-magnetic propulsion mechanism that is active and depends on their availability of energy. But you have an alignment or orientation of these cells that is passive and just depends on the interaction with an external field. So if you kill the bacteria, for example, they will still align with a magnetic field, but they will not move in that direction anymore. So you can think of them really as self-propelled compass needles. That's the picture I want you to have in mind. Okay, these are very small compass needles, so they are subject to fluctuations, and one can actually look at the alignment with a magnetic field as a function of the field strength. And what you see there, these are different measurements from the literature that we have just collected. What you see here is that the alignment with the field follows the Langevin curve. So this is just like the magnetization of a paramagnet. And different bacterial species have either different magnetic field strength, magnetic moment strength, or they have different fluctuations, because the fluctuations here are not necessarily thermal fluctuations. Okay, so we have a compass needle that for a field strength comparable to the magnetic field of the Earth, which is this line here, for most of these species gives a very decent alignment with the field. So they can really follow the magnetic field of the Earth. Okay, now you probably have the question in mind, why do they do this? Why do bacteria want to follow the magnetic field of the Earth? And to get into this, we have a look at the field of the Earth a bit more. So if we're somewhere here in the Northern Hemisphere, so I'm now in Central Europe right now, you see that the magnetic field of the Earth here basically has a component to the North Pole, but it also has a component that goes downward. So if the bacteria live in water, these bacteria live in lakes typically, then they experience a field that goes downward with some inclination, and that means they will also swim downward. And that's exactly what they want to do. They don't want to go to the North Pole. They are interested in the downward component, because that helps them to get to what's called the oxic-anoxic transition zone, which is their preferred habitat. That's a region where there is still some oxygen in the water, but if you go further down, there will not be any more oxygen. So you have a small oxygen concentration. Typically, that's in the upper part of the sediment, so it's kind of in the mud. And they live best where they have some oxygen, but not too much. Okay, this is something that we can look at in the lab. So Damien's group has done exactly this. They have a microscope that is built with these 3 pairs of Helmholtz coils, so you can precisely control the magnetic field inside. And then they put a capillary inside here. And this capillary is open at one end, so oxygen can come in from here, and it's closed at the other end. And they put in some bacteria. And what happens in that case is that these bacteria accumulate in a band. So you get some position here where you have a high density. You see this shown here. This is the meniscus of the water, and somewhere, some distance from the meniscus, you have this band which is dense in bacteria. So that's where they accumulate. And here you see the density profile plotted. In that case, this is very sharp. How it looks exactly depends a bit on the species that you use. And you can also see that there is an oxygen gradient. This was measured with an oxygen-sensitive dye. You see that this decreases pretty much linearly. And there is a low concentration, but still above 0, where the peak of the bacterial density is. Okay, we also can look at this formation of the band in a model. And what we do there is that we, in the simplest case, write down equations at the population level. We have 2 equations for bacterial density, bacteria swimming left and bacteria swimming right. They have their self-propulsion, and they have switching of direction from left to right and right to left. And then we have a 3rd equation for the oxygen. Oxygen diffuses in the system and is consumed by the bacteria. So basically, oxygen diffuses in, and where the big band is, the peak in density, that's where the bacteria consume all the oxygen. So there is no oxygen to the right. Okay, let's run this again, and you see that this indeed forms a band. It moves a little bit with the time until it reaches some steady state position. Okay, what I haven't told you about this model yet is how the directional information is used by the bacteria. And if we think about this, what kind of information do these bacteria need? Well, on the one hand, if you look at a bacterium here and one here, they have to swim into opposite directions if they want to get to the band. Okay, this one has to swim to the right, this one has to swim to the left. And that means they have to know what the local oxygen concentration is. If the local oxygen concentration is too high, they have to swim to the right. And if it's too low, they have to swim to the left. And then, of course, they don't know what left and right is, but rather they have to take one of the directions they can get from the environment. And there's two such directional quantities that they can use, either the magnetic field or the gradient of the oxygen concentration. Okay, so in principle, these switching rates for changing the direction, they depend on these three quantities: oxygen concentration, gradient of the oxygen concentration, and the magnetic field. And what we did in this simple model is that we simply said there is a step function in the oxygen concentration. So the right-left is higher if the oxygen concentration is low, and the left-right switching rate is higher if the oxygen concentration is high. And then we still have to see which of these two quantities the bacteria use, the gradient or the magnetic field. And in nature, of course, these give exactly the same information, because there will be less oxygen if you go downwards, and the magnetic field also points downwards. So this is kind of redundant information. But what we can do, and this can be done in the lab, is that we give them conflicting information. So we try to confuse them and see what they do. And this is an experiment that was done by Christopher Lefebvre. So he let the bacteria form the band that you see here. And then once the band had formed, he reverses the magnetic field. And he found out that what happens then depends on the species of bacteria that you use. So in one case, this is the Magnetospirillum that I have shown you before. You've seen this electron micrograph of. In that case, really nothing happens. The band just stays where it is. So that means these bacteria don't follow the magnetic field. That means they have to follow the oxygen gradient. So they really sense an oxygen gradient, follow the oxygen gradient, and that is dominant over the information they get from the magnetic field. And that means the magnetic field in that case doesn't really do very much. The only thing it does is basically project the motion down from a three-dimensional motion to a one-dimensional motion. So if they have to search by a random walk in 3D with the magnetic field, they basically have to search only along the axis of the magnetic field. So that means in that case, we could say these switching rates depend only on the oxygen concentration and its gradient.
Stefan, can I ask a question here?
Yeah, sure.
So this competition must be dependent on the strength of the magnetic field, or is it independent?
That's a good question. We don't really know.
Because in the lab, it should be possible to vary magnetic field and see when magnetic field overtakes.
Maybe I can come back to that later.
Okay.
So it's not something that we have looked at systematically, but I think there may be a way of even going against strong magnetic fields.
Okay.
Yeah, let me come back to this. If I haven't answered it by the end of the talk, just ask me again. Okay, there's still a lot of white space here on my slide. So that is because there are other cases, and sort of the extreme other case is in this bacterium, Magneto-coccus marinus. Once he switches the direction of the magnetic field, the bacteria move away in both directions. So they move to the left and to the right, which means that bacteria at high oxygen concentration, they swim in the direction of the field as they have before the turn. and at low oxygen concentration, they swim against the magnetic field as they have done before. So these bacteria really follow the magnetic field. That means they don't measure the oxygen gradient, or at least the magnetic field dominates over this. And the way we think about this is that they basically use the magnetic field as a proxy for the oxygen gradient. Because the magnetic field tells them how to go downwards, they don't need to measure the oxygen gradient to find where it goes downwards. And we can implement this by making the switching rates dependent now on the oxygen concentration. This we still need to get the difference between here, and the magnetic field as a directional reference. And if we implement this in the model, we have these two cases here. You can see in both cases the band forms, and now the magnetic field should switch. This one has switched, nothing happens. This one also switches, and you see two populations moving away in both directions. Okay, this model is kind of qualitative, is not a very quantitative model. So more recently, we tried to do somewhat better. And to do that, we came back to this active Brownian particles model that I have shown you in the beginning. So we made a very detailed model, and we in that case mostly means Agnese Corduti, a PhD student who was jointly supervised by me and Damien Fèvre. She made this model that includes the self-propulsion, so that's the active Brownian particle, the interaction with a magnetic field. So now we have a magnetic moment that gives rise to a magnetic torque in the orientation equation. But also, in addition, some active direction changes, which could be of two types, either tumbling, where you have a random reorientation of the cell body, or a reversal, where the bacteria just starts swimming backwards. And then she also includes chemotaxis or aerotaxis to measure the oxygen gradients. Okay, let me briefly walk you through these ingredients. So the active directional changes, in particular, the tumbling has the effect that if these bacteria swim along a magnetic field and then there is a tumble, there's a strong reorientation away from the magnetic field. And you see there's this strong change of the angle between the orientation and the field, and then a relaxation back towards the field. And then they swim along the field for a while, and there's another such reorientation. So, excuse me, there is basically a source of orientation fluctuation that is discrete, that comes in discrete events, and well, contributes to an average orientation that is not in the direction, not exactly in the direction of the field. That's shown down here, where you see that once you change the times between the tumbles, you get sort of different Langevin curves that look like there was a different temperature, but in reality, it's just that these tumble events have different frequencies. And well, while these Langevin curves can nicely be described by an effective temperature, if we look at the distribution of the orientation, so the full probability distribution of the angular degree of freedom, then you see if we take the effective temperature that describes the Langevin curve, that's this black line here, that is very different from what the model gives us as if we measure the distribution directly. So clearly this is not a thermal kind of motion, and describing this by an effective temperature is maybe just a, well, it's something you can do, but you shouldn't rely on too much. Okay, there is a question why axial means. So axial means that the magnetic field is not really followed as a direction, but it's still there as an axis. So it's this projection onto motion along one direction, but it can go both ways. Okay, there's another question. How does one define the effective temperature? Basically, if I take these curves, average orientation as a function of the magnetic field and the magnetic field strength, if this was a Langevin curve like in paramagnetism, I could say this should be dependent on mu times b over kt, and I can just take the T, the temperature in that expression, as a fit parameter, and that will give me some temperature that is different from the actual temperature, and I call this an effective temperature. That's very often used, but it's not always useful to do. So here it nicely works for this average of the cosine of the angle, but if we take the distribution of the angle, then this is not described by that temperature. Okay, and then the last ingredient is the chemotaxis or aerotaxis, which we just say that the run time, so the time it runs before a tumble or a reverse, this has some small value if it goes down the gradient, and then if it goes up the gradient, it has another value, and there is sort of a linear interpolation in between. And then if we include this in the mean square displacement, we go from these two regimes that we have seen before to having a third regime where we get this, again, directed motion on long time scales. Okay, that's the kind of model that we have. And what we now want to look at with that is what happens if we have two competing directions. We have a direction from a magnetic field and a direction from a gradient. And so what is shown here is that we vary the angle between the field and the gradient. And here at 0, basically both would point to the same direction. So they swim down the gradient or up the magnetic or in the direction of the field. That's both the same. And then we change that at 90 degrees, they're perpendicular. And then at the right end here, the two actually point in opposite direction. And what we see here is, well, if there is no magnet, if these bacteria are not magnetic, there is a certain velocity. And basically, the chemotaxis does whatever it does. They swim with a certain velocity down the gradient in that case. And whether we use tumbles or reversals, that doesn't make much of a difference here. That's the solid line and the dashed line. Once we include a magnetic moment, then at low angles, so if the field and the gradient point in the same direction, then we get some speed up from the magnetic field if we have reversals, and even more speed up if we have tumbles. But then if you change the angle between the field and the gradient, you see that eventually with the tumbles, this goes through 0 and then becomes negative. So with the tumbles, basically these bacteria will follow the gradient, the magnetic field eventually, instead of following the gradient. So in that case, maybe this also is a partial answer to Debashish Chowdhury's question. In that case, the field does essentially override the gradient sensing. And of course, this would be more so if the field is stronger, and maybe less so if the field is weaker. And eventually, if the field is very weak, then they could still, by fluctuation, swim in the opposite direction. However, with reversals, this angle between field and gradient also decreases the speed, but it never goes down fully to 0. There's still a positive speed at 90 degrees. And then because they can swim backwards also, and just as forwards, this speed actually increases again if the angle goes towards 180 degrees. So what we conclude from that is really if these bacteria should be able to swim against the magnetic field, they should not do a run and tumble such as E. coli does it, but they should have the option to swim backwards, which means basically they have the option to swim against the direction of their magnetic moments. So they don't have to reorient their cell body. They can keep the cell body aligned with the magnetic field while still swimming against the magnetic field. So that's important for them to do. And if they can do that, then the field strength is not so much of an issue because you don't have to overcome the field by fluctuations, while in the case of tumbling, you have to overcome the field by fluctuations because the cell body needs to be oriented against the magnetic field to swim against the magnetic field. Okay, let me come back to the band. So we used then this model also to look at the band formation, and mostly what we had to do here is adjust the reversal rates. So we need reversal to get this band forming, because I mean we would need here swimming in the direction of the field and down here swimming against the direction of the field. And once we adjust the reversal rate, we can nicely quantitatively find agreement with the experiments. For example, here you see the position of the band, which starts very close to the meniscus and then moves a little bit towards the interior of the capillary. And on the right-hand side, you see the density profile in the band. So the dots here are experimental, is the experimental density profile, and you see this line from the simulation. So there's very nice agreement, and we can quantitatively describe the formation of this band with this active Brownian particle model if we have included reversals and chemotaxis, or aerotaxis, chemotaxis for oxygen. One question that I always got when I talked about this is what happens if you have interactions, if you have many bacteria? So in most cases in the experiments, actually the density is relatively low. So interactions, magnetic interactions between the bacteria probably don't play much of a role. But of course, from the physics point of view, that's an interesting question. So we thought, well, let's look at this. We now have many active particles, magnetic particles, or dipolar particles in general. Let's see what happens if they interact with each other. And we know that if we have passive particles, let me start this. This is actually starting already. If we have passive particles, then we know they form chains. If you have magnetic beads like these, they form chains, and they can also form rings. And we have actually had a look at these earlier because we were interested in the formation of this chain of particles inside the bacteria. But now the question that we had is, well, what actually happens if we make these active, if we have now active particles with a magnetic moment? And well, what happens is what you see here. We have a few active particles here in a spherical or circular chamber or confinement. And you see they form chains, and these chains move, but occasionally they also form rings. So this one here forms a ring, and then the ring just rotates, and it doesn't move in a direct fashion. Here you see that they also form rings. They tend to form rings when they collide with the wall of the confinement. So this looks like an interesting problem, and we had a look at this. This is actually, I should say, this is the work of Vitaly Teletsky, a current PhD student in my group. And what he did is that he quantified the chain versus ring behavior by looking at this parameter C. So for C, what he compares is the distance between the ends of a chain, so the end-to-end distance of a chain, and the contour length of the chain. So this L is this length here, and D is the distance between the first and the last particle in a chain. In that case, they are the same. In this case, they are very different. And then the parameter C is basically just 1 minus the ratio of these two. So that when this is 0, this means we have a chain, a straight chain, and if it's 1, we have a ring. And he varies the parameters in simulations. This is the velocity, and this is the magnetic moment of these particles. And for all parameter combinations, he takes a histogram over this parameter C. And what you see is that in many cases, this has a peak at 0. So it's dominated by chains. But there are also some cases, in particular in this upper right corner here, where this is dominated by a peak at 1. So that means it's dominated by rings. And then you can use this and basically take all this information and make a kind of a phase diagram. It's not really a phase diagram. Let's say a configuration diagram here. And you see there is some region where most of the particles are not in structures. So they the interaction is too weak to really get them into some structure. There are areas where you see mostly chains, and then there is this big area here that is dominated by rings. And one of the interesting aspects here is that this is very much dependent on the boundary conditions that you use in this. I have already shown you in the movie that there was one case where we had this chain that collides with the boundary and then forms a ring because it bends at the boundary. And if you look at this plot here, you see for a quadratic box and for a circular volume here, you see the C value plotted locally, and you see that mostly you get these rings close to the boundary. So this seems to be a strong influence here. And indeed, if we look at these phase diagrams for different boundaries, so this is the quadratic and the circular case, and then in that direction we also vary what happens exactly when they interact with the wall. So this alpha parameter basically controls a torque that the wall might exert on them. So the simplest case, alpha is 0, the wall doesn't exert any torque. You already see that there's a difference here, but that gets even bigger. And it tends towards favoring rings, the chains over the rings here. So this ring area, you see, gets smaller if the boundaries are less driving the ring formation. Okay, so this is very different from what you have in thermodynamic equilibrium, where in principle the boundary conditions shouldn't matter that much. Here they do. And it would be even more extreme. I don't have it on the slide. It would be even more extreme if we use periodic boundary conditions, because then no such collisions happen and the rings are even less likely then. Okay, at this point, I want to change gears a little bit. So far, I've mostly talked about these bacteria at a very coarse-grained level. We say the bacteria, basically, they're just point particles or little spherical particles that have an orientation. So they have a direction in which they go and they have a magnetic moment. But of course, they have much more detailed structure. And if we want to look at how they move, then we have to look at that more detailed structure. And that's what I want to do in the last part of my presentation. And to that, I want to come back to this slide that I had at the beginning, where I've shown you that we have motion, active movement in life on all scales. And what I want to draw your attention to at this point is that if we have motion at these different scales in life, the physics of that motion is different on every scale. So on the meter scale, the dominant physics is inertia. On this micrometer scale of cells, we have friction and viscosity as the dominant physics. And then if we go to the intracellular scale, we have Brownian motion as the dominant physics. So even though we have motion on all scales, we have very different physics. And again, we look at the middle case here. And in terms of hydrodynamics, because we're talking about swimming, this is high friction case is the case of low Reynolds number hydrodynamics. And if we have hydrodynamics that is dominated by viscosity, there are two important properties of that hydrodynamics, namely it's time reversible and it's linear. So velocities are proportional to forces. And one consequence of that is the so-called scallop theorem that says that reciprocal motion does not result in net propulsion. So the hypothetical scallop of Purcell is this double shell thing, and the shell opens and closes. And when it opens, it will move in one direction. And when it closes again, it will move backwards exactly in the same direction, exactly the same distance in the opposite direction. Okay, so this can only lead to back and forth over a little bit, but it doesn't need in any net propulsion. So we need other mechanisms at low Reynolds number for propulsion, and one of them is this rotation of a helical structure, such as the flagellum of a bacterium. And if we want to look at this for our magnetotactic bacteria, We can do this by making a more detailed model where in the simplest case, we take a cell body, for simplicity again a sphere, and we attach one flagellum to it, which is basically a helical filament that is discretized into a chain of beads. And then we write down these linear equations of motion where the velocities and angular velocities of all these particles are proportional to the forces and torques acting on them. And the proportionality is given by a matrix that contains all the hydrodynamic mobility coefficients. So these have been calculated in past. There are different approximations for them. The one we use here is mostly the Rodney-Prager approximation. But basically what is important is that we have some contributions in here that couple rotation and translation. So if we rotate the flagellum, this results in the translation of the cell body. And the forces and torques that we include here are, of course, the magnetic torque that orients the cell body, but then also elasticity of the flagellum and the motor torque that rotates the flagellum. And this gives rise to a large set of equations of motion that I don't want to go into in detail now. What I want to show you is how this looks in the simulation. Here you see our magnetotactic bacterium. This is the direction of the field. And you see on the one hand, it propels. On the other hand, over some time, it will orient in the direction of the magnetic field and eventually swim in the direction defined by the magnetic field. And I should mention that all these simulations here, they were done by Sarah Mohammadinejad, who is a postdoc in my group. Okay, now the bacterium disappears and we should probably move on. This was a kind of a caricature of a magnetotactic bacterium, spherical cell body and one flagellum. We wanted to look at this for one specific realistic case, which is the Magnetococcus marinus. Maybe you remember this was the one that in the case of band formation after reversal moved away in both directions. Okay, this is the one, and it has an almost spherical cell body, and it has not two flagella, but two sheathed bundles of flagella. So these are bundles that cannot come apart. So they are effectively like two flagella, and they are approximately on the same side of the cell body. So you can imagine it like this. And the magnetic moment has some strange angle, relatively large angle with these flagella. So this is a relatively unexpected and strange geometry. So what we expected and what everyone else expected is that they swim like this. They have the two flagella in the back and they basically swim essentially straight, at least as long as we don't have a magnetic field. Once we have a magnetic field, then because the magnetic moment is not parallel to the direction of motion, then they might start swimming on a helical trajectory around the magnetic field as this magnetic moment precesses around the direction. But we first wanted to look at the case without a magnetic field. And Klaas Bente, who was a postdoc in Damien Favre's group, he actually looked at this experimentally by doing three-dimensional tracking of these bacteria. And what he found, these are many trajectories, and let's look at one in more detail. What he found here is that these bacteria, in the absence of a magnetic field, move on helical trajectories. So they make these helical trajectories here, and then occasionally they make very abrupt turns in that helix. And I should say, without the three-dimensional tracking, it would be very difficult to see this. So we have these nice helical trajectories and abrupt turns. If we look at the speed, which is plotted up here, they are very fast. So they go at maybe 300, 400 micrometers per second, which is 300 to 400 times their body length per second. Incredibly fast. And once they turn, you see the turns denoted by these red dots here. Once they turn, you see that the speed goes down to 0 for basically just 1 or 2 data points, okay? 1 or 2 data points is 5 milliseconds. So they turn also very fast. That means these bacteria get 2 gold medals, one for the speed of swimming and one for the speed of turning. Okay, that's nice for them, but for us, we have these helices that we need to explain because we didn't expect them. And in fact, Klaas and Sarah tried a lot of things. One of them was actually to see the flagella, which we don't in the experiments. We don't see the flagella directly. So he tried to get higher resolution microscopy to see them, tried to label them somehow, but most of these things didn't work. And on the other hand, we used the simulation to see what we could make asymmetric between these flagella to generate a helical trajectory rather than a straight trajectory. And well, it turned out in the simulation, the one thing that can really generate these helical trajectories is if the 2 flagella rotate in opposite direction. In that case, the bacteria will swim like this. So 1 flagellum in the front, 1 in the back, and the cell body rotating around, and this gives rise to this helical trajectory. I'll show you this in the movie. So here it swims 1 flagellum in the front, 1 in the back, and the cell body rotates around it and thereby makes this nice helical trajectory. Okay, if we look at it with higher resolution, then one can see, this is experiment, this is simulation. What you see is that this helix really is not 1 helix, but it's 1 helix on top of another helix. There's a question, is the helicity due to the torsional degrees of freedom? I'm not sure. I don't think so, but of course you need a flexible flagellum here to make it move in the front and in the back. Otherwise, they would just be straight and parallel. I don't think that the torsional degree of freedom is really what you need. Okay, but what you see here is you have this 1 helix on top of another helix. Some of that has to do with the attachment to the cell body, which is the helix is usually not attached on its axis. So that will give rise to the smaller helix. And then the larger one is due to this 1 being in the front and 1 being in the back. Okay, I said already, experimentally, unfortunately, we can't see the flagella. So this is the highest resolution video that we have of the motion in the experiment. And what you see here is, well, there is this white dot. There's a bright spot on these cells, and you see this bright spot rotating around the cell body, which is reminiscent of what we see in the simulation. Klaas believes that this spot is where the flagellum is really attached to the cell body, but of course, we cannot prove that. So we see a rotation of the cell body that is consistent with what we see in the simulation, but we don't have full proof of this type of motion. There's one more question. Are the flagella located on the opposite poles of the cell? No, they are not. That's one of the things that makes this complicated. Let me just go back. So they are kind of on one side of the cell. So you need some flexibility to bring them 1 point to the front and 1 point to the back. If they were on 2 poles, there are other bacteria that have them at 2 opposite poles. That, of course, would be much easier. Move back here. Okay, so everything I've shown you so far was without a magnetic field. So if we now add the magnetic field to this motion, then what happens is what we would expect that happens, namely that this helical motion that we had so far gets now superimposed on an even larger helix that comes from the precession of the magnetic moment in the magnetic field. So now we have basically 3 helices, a smaller helix on a larger helix on an even larger helix. Okay, and we see this both in simulation and in the experiment. Okay, I think my time is basically over. I have 3 slides about micro robots, which I think I will not go into in detail. It's also not so directly related to the bacteria, but I just want to mention that, of course, people are interested in making use of what we learn from the swimming of bacteria in developing synthetic magnetic swimmers. Magnetic because we can use magnetic fields for remote directionality, which is very useful. And then the question is, how do we propel them? And there's different ways. One of them would be to take a bacterium and functionalize it. So you could attach it to whatever structure you want to propel, or use a bacterium to transport cargo. This is something I don't have results on now, but we're working on this both in the simulations and in the experiments. And another thing is that you use completely artificial structures, for example, helical structures that you can propel. And well, if you have, for example, a helix that has a magnetic moment perpendicular to the helix axis, then that's something that you can propel. This is from a simulation, and people have done this already. Maybe 10 years ago or so. This is one example from the lab of Per Fischer in Stuttgart, who made these very elaborate helical structures that can be propelled now with a rotating magnetic field, because you generate now the rotation with a magnetic field in addition to the direction. One thing that we have been asking over the last years is, do we really need a helix, or can we use some other structure? And the approach that we have used is to make random structures, and among the random structures, select those that swim well. I don't want to go through this in much detail, but you can take this structure here, which is not quite a helix. You could imagine it's a small piece of a helix, maybe. This one is one of the fastest ones. It's comparably fast to these synthetic structures that people have made. And of course, if you have random structures, you want to have a way of generating them systematically, because if you make them randomly, you will only make them once and you cannot reproduce it. So the way is that if you have such a structure made randomly, identify this as a good swimmer, then you want to reproduce this in large amounts, for example, with 3D printing. And where we are right now is that we have control. Well, the experimentalists have control over making these structures. What they don't have good control over yet is the magnetization of these structures. This is what you see here, the swimming velocity, in that case, over the rotation frequency. This differs very much between different realizations because there is no such control over the magnetization yet. So that's another step that needs to be taken. Okay, with that, I'm at the end of this presentation. What I try to convince you is that magnetotactic bacteria provide really a nice model system with which we can study the role of physics in biology. Because we have a physics that is relatively simple. We have magnetic forces, magnetic orientation, and it has a role for the biological function of these organisms, but it also provides some constraints. For example, you can't swim against a magnetic field, at least not easily. You need specific mechanisms. For example, you need reversals instead of tumbling for directional changes. So the magnetism provides these cells with new functions, but it also puts them under new constraints. And well, these functions, importantly, directionality and remote control, if we're interested in a function as someone who wants to apply them. I briefly mentioned that they provide inspiration for these biohybrid and synthetic microrobotics. And maybe one point that you have seen from this strange Magnetococcus case that apparently swims in this weird way is there is really a diversity of propulsion mechanisms even within bacteria that have flagella. So there is this run and tumble of E. coli, which everyone takes as the standard model, but there are many bacteria that behave differently. And that was one example. And there are many more that have different flagellar arrangements, different coordination mechanisms between the flagella. Okay, so there's still a lot of things to be studied here. One important aspect that's not mentioned on the slide, but that was kind of implicit in the whole talk, is that active motion, energy consuming behavior, is the key for everything of this to work, for the rotation of the flagellum, for the formation of these bands and so on. So that biological systems are active systems is one of the key properties that we need to consider here. And finally, if you want to read more about this, we have written a long review article last year here in Physics Reports, where you can read much more about magnetotactic bacteria and also a bit about the biohybrid and synthetic micro-robots. So finally, I want to thank the people involved, in particular my group and the group of Damien Fèvre. There's a list here. I already highlighted Agnese Corduti. She's here in both sides of the list because she was a joint student in our groups. And then I mentioned Vitaly Teletsky, who did the simulations of the interacting magnetic particles, and Sarah Mohammadinejad, who did the simulations of swimming with the detailed model of the flagellum. I also should highlight some of the experimentalists: Christopher Lefebvre, who did the original band formation experiments, Klaas Bente, who studied the three-dimensional motion of the magnetococcus, and Peter Fach and Felix Bachmann did the magnetic propellers, these micro-robots that I briefly showed at the very end. And of course, Damien Fèvre, my long-term collaborator in this project. And I also thank our funding sources. And I thank you very much for listening, and I'm happy to take more questions.
Thank you, Stefan, for the wonderful talk. So session is now open for the questions. So you can either unmute your mic and ask the question, or you can type the question in the chat box.
There's already one in the chat box.
Yeah.
In the slide where you were showing the effect of the geometry on the boundary of the ring and chain structures, what will happen if you consider a spherical surface? You mean if we confine them into a real sphere?
On the surface of the sphere.
I don't know. We haven't tried, but I would expect that it's similar to the circular structure. So the main difference between the circular structure and the quadratic structure is that you don't have the corners. So once a chain gets into a corner, then it's likely to get trapped in there. And once it's trapped and basically stays in this corner where it has two walls to interact with, then it's likely to form a ring. And I think if you go to a three-dimensional corner, that would be even more so. So I think you would have a similar effect, that basically in a cubic box, the rings would be more likely than in a spherical. Second question, how is the flagellar length? How does that affect the swimming properties? Have you looked into mutants? No, we have not looked into mutants. One of the problems with these magnetotactic bacteria, as nice as they are, is that it's very difficult to do genetics with them. So making mutants systematically is hard and is only possible in some species. I don't think anyone has looked at flagellar mutants. We have looked at it in the simulations, and the length, the length had an effect, but I don't remember. It's nothing spectacular. I think they get a bit slower when the flagellum is longer or so. So there is a quantitative effect, but it doesn't change the qualitative behavior.
I have a related question, Stefan. If you change the length, so maybe it's possible that the pitch of the three helices, they become commensurate or incommensurate, and that can probably have some significant effect?
That might be possible, yes. Yes, I mean the parameters of these helices depend on these geometric properties of the flagellum to some extent. So maybe yes, that's something we could look into. Yes.
Okay, thank you.
Someone is raising their hand.
Yeah, Cecile.
Cecile.
Hi, Cecile.
Yeah.
Hi, Stefan. Thanks for the nice talk. I had a very naive question about your introduction. You were mentioning that bacteria were sensitive, were interested in the component of the Earth's magnetic field that goes down, but still they will feel the one going to the north. And how is it that they don't, that they are not all stuck in the north of the lake or?
I don't think anyone has looked at that. Yeah. I don't know. Maybe when they swim upward along the same direction, they would also go south. So if they use the axis and they go upwards.
Okay, some kind of averaging that would lead to zero.
That's what I would guess, but I'm not sure.
Okay.
I don't think someone has looked at it. I mean, the bigger puzzling question is actually what do these bacteria do at the equator? Because at the equator, the field is completely horizontal. So you would assume that there shouldn't be any magnetotactic bacteria there, but there still are. And I think that's an unsolved problem.
Uh-huh. And my another question was, at the beginning, you were showing some fluctuations around, well, how well they follow the magnetic field. And you had differences between species. But
Yes.
then you have strong differences between individual bacteria, or is it the same for all of them?
So no, there are difference between. So if you take bacteria of the same species, there is a certain distribution of their magnetic moments. So that's one thing. So some are more magnetic than others. That would give rise to differences. There are also, at least in some cases, we have seen that there's big difference in the velocities. So that velocities also differ from cell to cell. That would not affect the alignment, but yes, so these are really not all exactly the same. So I would say the heterogeneity in the magnetic moment is probably the biggest contribution in terms of the alignment.
Okay.
So of course, if the fluctuations are not thermal, but something that comes from inside the cells, then that might also not be the same for all the cells.
Yeah. Okay, thank you.